Roughly 50,000 years ago, human behavior underwent a severe acceleration. Populations across the globe began crafting composite tools, burying their dead with formalized rituals, and producing symbolic art. This chart tracks hominid grain volume against time. Notice our ancestors achieved modern brain volume roughly 300,000 years ago. Yet the leap in cultural complexity is clustered entirely at the 50,000-year mark. That leaves a 250,000-year gap of relative behavioral stagnation. This timeline forces a specific question: what exact mechanism catalyzed our modern behavior? The fossil record cannot resolve this discrepancy. Instead, researchers are decoding the chemical code of our ancestors. Recent high-resolution DNA sequencing has fractured the linear models of how we developed. Currently, two highly divergent models are attempting to explain this new genetic data: the mainstream consensus of biological gradualism and a fringe theory proposing a deliberate genetic trigger. To understand our origins, we have to examine the exact same genomic anomalies through both of these fiercely opposed frameworks. Modern genome mapping has revealed unexplained, highly divergent sequences buried within our DNA, distinct signals introduced roughly 50,000 years ago. This diagram maps a phylogenetic tree containing a ghost lineage, a distinct hominid group leaving zero fossil evidence. We know they existed because genetic echoes remain intact in our code. These populations lived alongside early Homo sapiens, passing genetic material into our lineage. While inherited sequences provide the raw material, biological function depends on the mechanisms that control how that material is expressed. This brings us to a specific set of genomic sequences known as Hakers, human-accelerated quick-evolving regions. Hakers do not encode proteins. They operate as high-speed evolutionary switches, determining exactly when and where other genes are turned on or off. These regulatory switches are disproportionately responsible for directing the development of the central nervous system and the physical structure of the brain. These two verified anomalies, ancient ghost DNA and hyper-accelerated regulatory switches, form the foundational data that any model of human origins must now satisfy. The mainstream biological consensus interprets these anomalies through evolutionary gradualism. Biologists have discarded the concept of a missing link. There is no single linear progression of transitional species. This animation shows the outdated ladder of evolution shattering to reveal the modern consensus, a weakly structured stem. Our history is a messy, interconnected network. This web allowed for continuous genetic exchange, including documented archaic intergression from Neanderthals and Denisovans. This constant networking directly impacted physical brain development. This 3D volumetric model compares historical scaling of the primate neocortex against the cerebellum. It refutes the assumption that cognition is driven solely by a large neocortex. The cerebellum and subcortical structures scaled simultaneously. Brain development was a mosaic process. This mosaic pattern applies down to the genetic level, specifically to FOXP2, frequently and incorrectly labeled as the singular gene responsible for human language. The human-specific variants of FOXP2 did not arrive via a sudden mutation. They result from cumulative selective sweeps operating over hundreds of thousands of years. Under mainstream interpretation, the cultural explosion 50,000 years ago was a tipping point, reached only after millions of networked biological shifts finally achieved critical mass. The interventionist framework looks at this exact same data and reaches a radically different conclusion. Proponents argue from statistical improbability. They contend that multiple simultaneous regulatory shifts, like those seen in AHQRS, are highly unlikely to occur purely through random mutation. This animation models the primary physical anomaly cited in the interventionist framework. Two separate ancestral primate chromosomes shift, align, and fuse end-to-end to create human chromosome 2. Interventionists argue that the precision of this fusion, which streamlined the human karyotype while perfectly preserving utility, bears the signature of intentional tampering. This perspective also reinterprets the 50,000-year-old West African ghost DNA. Rather than viewing these ghost lineages as natural evolutionary contemporaries, this framework proposes they were specifically engineered genetic stocks introduced into the population. Interventionists point to the structural complexity of the Great Ziggurat, anchoring the historical weight of Sumerian civilization, a culture that recorded specific narratives about our origins. Sumerian texts describe external entities, the Anunnaki, splicing their genetics with local hominids to create a worker race termed the Adamu. The interventionist model concludes the cultural leap 50,000 years ago resulted from an external genetic trigger designed to bypass millions of years of gradual evolution. These two frameworks process the same biological anomalies through entirely different philosophical lenses. Mainstream science relies strictly on Occam's razor. It favors naturally occurring, observable mechanisms like introgression and genetic drift, no matter how chaotic the resulting timeline appears. The interventionist approach answers the statistical improbabilities of the genome by introducing a massive assumption: the existence of highly advanced external architects. Regardless of the chosen framework, the linear model of human evolution no longer accounts for the genomic data. Discoveries of 50,000-year-old ghost DNA and high-speed regulatory switches replace the 19th century focus on gradual anatomy with a 21st century map of genomic complexity. The paleogenetic record proves our origins are a deeply networked system of archaic mixing and rapid regulatory shifts.
The recent findings released by Project Pursue have fundamentally reinvigorated the interventionalist argument in human evolutionary theory by definitively placing an advanced intelligence within our local vicinity during critical periods of hominid development. For decades, the interventionalist hypothesis, which posits that sudden human biological and cognitive leaps were guided or accelerated by an external entity rather than strictly by unguided natural selection, lacked the empirical anchor of a confirmed non-human presence. By verifying that a highly advanced intelligence was active in our area at the exact time our ancestors experienced unexplained evolutionary anomalies, the Project Pursue data finally provides a plausible mechanism and a prime suspect for these rapid changes. Consequently, sudden expansions in hominid cranial capacity, abrupt genetic shifts, and uncharacteristic technological advancements in the archaeological record are now being seriously re-examined, not merely as evolutionary flukes, but as potential evidence of deliberate, localized intervention by this newly confirmed intelligence.